Hey everyone, we're going to look at the cytology and endomembrane system introduction here. So hopefully you have the PDQs that go with this. Okay, so I'm going to go ahead and start off in full screen here. Here we go. So when we're looking at cytology in the endomembrane system, we are basically going to try to answer the following question. Why does life require cells? So what's the whole point of them? How are they organized, and what is the advantage of having organelles? And how do the interactions of cellular components allow for life processes? This is sort of a tour of the cell part one. You can kind of think of it that way. So I know if you've had Mrs. Fellows, I think she probably did a whole spoof on dead white men and their prevalence in science throughout the ages. And again, discovery of cells, again attributed to another dead white man. And we have Leeuwenhoek and Robert Hooke that are main contributors, but they're not the only two contributions. Cytology, just so you know, cytology means study of cells. So if we're looking at cells and we're researching them, experimenting, looking at them through microscopes, we are basically following along cytology. Now, the cell theory is that all living things are made of cells. Number 2, cells, basic unit of life. And number 3, all cells come from pre-existing cells. So Leeuwenhoek was the first person to look at biological material under a microscope. So he was the first to really use a microscope. And then you had Robert Hooke, who kind of coined the term cells. And he coined them from the appearance of cork under a microscope. And it's these like little, to him, it looked like little openings under a microscope. Now, there's different techniques that we use, microscopy, different forms of it. We have light, which is like what you're used to using with the standard microscopes that have the light that come up from the bottom and it has the little tray on it. Maximum magnification is about 1,000 times. Maximum resolution is 10 micrometers. Let's see if this goes in a little bit. Okay. We have different ways that we could do it. We can fluorescence it, and that's with different radioactive tags that are given to different structures, that tag different structures. Phase contrast, this is mostly what we use right here. And then we can also use stain to help identify them. We will look mostly at phase contrast and bright field stained appearances. Sometimes we'll look at unstained specimens, but these 3 right here in our scope in the high school level and even in the college level for the most part, unless you go into research, will sort of work with these 3. There's also electron microscopy. Electron microscopy involves either transmission electrons or scanning electrons. Very expensive microscopes. You'll use them in research, but they can get much more detailed. So benefits and problems of the 2 different types. Well, the light microscope's cheaper, easier to access. Resolution's not as high. The electron microscope's much better resolution, much greater magnification, but also much more expensive for the technology and it's not as easy to access. So unless you're at a high-grade lab or a university research institution, you probably won't have access to a scanning electron microscope. Now, sometimes you need to separate cells in order to look at them. Oopsie. And so we have cell fractionation. It's a way to isolate different components of the cells for detailed study. And so if we have a bunch of cells, but we only want to look at like the mitochondria, for instance, we do fractionation. And so what that is that we just blend it up super fast, and then it kind of breaks it apart, and then it settles out depending on the density of the different organelles that we're looking at. Okay, so we blend, we have this whole mixture of stuff, and then we have these little pellets, like the pellet is going to be rich in nuclei and cellular debris, and then we pour off the supernatant and leave the pellet, and we do it again. We centrifuge it, spin it around, let the pellet settle now, and now this pellet has mitochondria in it, and then we keep repeating the process. Now, there are different types of cells that we're going to go into, and in a way they sort of relate to Cell size. Cells exist in the 10 micrometer to 1 millimeter size range. So 1 millimeter is actually quite big. And so the smallest one, if you look at atoms down here at 0.1, the smallest molecule is 1 nanometer here. And then you have your biomolecules, these molecules that make up the cell here, make up living things. So we have our lipids and proteins. We have our ribosomes that are going to process the DNA to make the proteins. We're going to have like our carbohydrates and whatnot in here as well. And then we have viruses. And then we have our smallest bacteria. Okay, the smallest bacteria, you can see it with electron microscopes. We really won't be able to see it with our light microscopes unless we have a super expensive one, a super resolution. Right in this range here, you have most bacteria. Most plant and animal cells are going to be a little bit bigger than bacteria. You'll be able to see some bacteria with a light microscope. It's kind of difficult to differentiate, though. Bacteria is a little bit harder to see unless it's stained really well. We will be able to see plant and animal cells with our light microscopes. That's not a big deal. And here we have human egg, frog egg, chicken egg, and then we even have some length of some nerve and muscle cells. Nerves and muscle cells, because of their design, because of their ganglia and their dendrites, they can actually be quite long in the body, and some of them are. Okay, and so this one right here is probably going to be your largest size cell. There are physical constraints on cell size. So cell size lab that we do, we don't, hopefully found out that there are constraints on the whole exchange of matter. Now, we don't want a cell so small that we can't fit everything inside, but we can't make it too big because we need to be able to exchange the matter with the environment efficiently. And so this would be too small to fit everything in, maybe. This might be too big. But if we can compartmentalize, if we can have lots of different places that we can increase the surface area, then we can get everything in, but keep it as efficient as the small ones are. And so when you looked at the agar cubes, that's the whole idea here, is looking at constraints and limits to cell size. Now, there are two cell types. There's the prokaryotic, simpler ones before the karyote, before the nut, or before the egg, before the true organelles. They're smaller, and they're much more abundant. And so we're looking at the bacteria for the most part here. Okay, so hopefully you remember this picture from like general biology. Eukaryotic cells are ones with lots of membrane-bound organelles, larger, and there's two major types: plant-like, photoautotrophic, and animal-like, chemoheterotrophic. Okay, photoautotrophic, it's able to make its food from light. This one, it makes its energy from other organisms, other substances. Protocells fall under plant-like cells. Fungal cells fall under animal-like cells. Okay, two major points here. Why the heck do we have so many organelles? And it kind of goes into that idea of increasing the surface area to increase the efficiency. Well, now we're still increasing the efficiency, and we're doing so by having different organelles do different jobs. So it's all about efficiency. Both areas, surface to area volume is about efficiency, or I should say cell size is about efficiency in one form, and this is about efficiency in another. So why organelles? Whoopsie, by enclosing parts of the cell and membrane, eukaryotic cells are able to specialize. Membranes isolate different areas of the cell, which allows the cell to have different conditions in different regions. And so this is really going to play an effect when you're looking at enzymes. And remember, different proteins have different functions and can only exist at certain conditions. If the conditions change, that protein may be denatured. That shape, because the shape will change, the pH will change it, the temperature will change it, etc. And it will no longer be able to function. So it has to stay at a very specific pH value. And so different organelles allow for these different pH values to exist. The membranes also provide surface for various reactions. So when we're looking at respiration and photosynthesis, and we look at like The electron transport chain, for instance, it actually utilizes the cell membrane and different gradients and especially this concentration of different molecules in order for it to actually produce and make the molecules that's needed or break down the molecules that it needs to. The specialization of cellular regions is what makes eukaryotic cells so much more complex. They can do so many more things. They can utilize so many more molecules that would normally be found in different environments because of this use of organelles. And it's a prerequisite for multicellular life, so they can start doing then different jobs. If you're going to have a multicellular organism, then these organisms are going to start partitioning the jobs that need to be done. They won't be able to do everything within one cell. So many compartments, so many options. I hope this looks familiar. As a reminder, all cells must do the following to stay alive. Process matter. Take it in, acquire it, make it, synthesize it, break it down, digest it. Process energy. Energy must be provided to do all this stuff. It comes from one or two places, so think autotroph, heterotroph. Where would these come from? Processes information. So there's instructions for how to process this matter. And then, of course, we need to reproduce and pass this information along and communicate with other cells. And cells have systems to do all these things. This is in another Prezi. So proteins. Proteins are the molecules that a cell uses to do most of its work. I call these the minions of the cell. Proteins do everything. It's like Gru telling the minions to go do the bidding of the evil, the evil taking over the world type stuff. And then the minions just go out and do it. And that's what proteins do. They build things, they build molecules, they break them down, digest them, they carry out chemical reactions, they receive and send messages. We previously discussed protein structure, and the instructions to build them are kept within the DNA. The nucleus, the instructions to build proteins are stored in DNA. We call them genes. And so each strand of DNA is going to have different genes on it, and the genes are the sections of the DNA which code for the making of a particular protein. Now, the nucleus is a double membrane bound organelle with protein pore channels. Now, what this protein pore channel is allowed to do is select information to leave the nucleus. Only select, because this protein is only going to recognize certain molecules to leave it. It's the site of DNA storage, but it's also the site of replication. And so here you can look at little pores in magnification from the electron microscope. Ribosomes are found in the cell. They're a complex of both RNA and protein. There's two units. There's a large unit and a small unit, and they come together, and they are sort of the factories where these proteins are made. They read and decode the message at the RNA, and the tRNA will bring in the appropriate amino acids and build the polypeptide chain. Now, when we look at nucleus, we oftentimes see the nucleolus, that darker spot inside the nucleus. This is where ribosomal RNA genes are concentrated. So to make this ribosome right here, that's where the genes to do that are found. Okay. Now, how eukaryotic cells send proteins from ribosomes to their destination can be found through the use of different organelles. So there's two kinds of ribosomes. First off, there's either free ones that are floating around in the cytoplasm. Those mostly make proteins that actually stay within the cytoplasm. They don't go anywhere else. But if you need to make a protein, if a protein is needed outside of the cell to be exported from the cell, it will be made at a bound ribosome, and those are attached to the ER. So when you're looking, hopefully remember ER, endoplasmic reticulum, there's smooth and there's rough. The rough ones are studded with the ribosomes that are making the proteins that are going to be exported from the cell. So here you can see some free ribosomes, and here you can see the ribosomes attached to the membrane. Now, the endoplasmic reticulum is just like a highway almost. It's a network of membrane-bound channels attached to the nuclear membrane. There's rough with the ribosomes on it, and then there's also free ones further away, and the free ones are mostly creating and transporting lipids. That's a big job of the smooth or the free ER. We also have little vesicles. Vesicles are just like a little bubble that surround molecules to help it transport through the cell and leave the cell if necessary. And then we have a Golgi apparatus. If a protein is going to leave the cell, or any molecule is going to leave the cell, it must be processed, packaged, and tagged in the right way to leave the cell. And that's what happens at the Golgi apparatus. And so here's an example of the Golgi. And then we've mentioned before, but we have plasma membranes. The plasma membrane is a phospholipid bilayer. It has embedded proteins to help molecules go through. It's got proteins to help recognize other molecules. It's got proteins to help recognize other cells. The carbohydrate side chains will help with the cell-to-cell recognition. But the whole function of the plasma membrane is to control what goes in and out. So these organelles make up what's called the endomembrane system. They all have something to do with having a membrane, the use of their membranes, etc. If there are any questions, please let me know.